This method can help answer key questions in the field of infection and immunity, such as how do individuals respond to different routes of infection?
The main advantage of this technique is that it allows for the examination of gut immunocompetence, which plays a significant role in host defense against infection that are mainly acquired through feeding.
This method has the potential to be used in species other than Drosophila and can be used to study other pathogens as well, such as viruses.
Generally, individuals new to this method will struggle because of the required experimental rigour during these microbiology protocols.
Maintain the flies in 23 milliliter plastic vials containing 7 milliliters of freshly made Lewis medium. To plug the vials, use non-absorbent cotton wool. Keep them at 25 degrees Celsius under a 12-12 light-dark cycle with a relative humidity of about 60%. Every 14 days, transfer 20 to 30 adults to a new food vial, with a dash of dry yeast added to the surface. After 2 to 3 days, dispose of the adult flies if sufficient eggs have been laid. To collect eggs, prepare a population cage. First, add water-soaked cotton wool to provide moisture. Then, for a substrate, use a 75 milliliter plate filled with apple agar and a spread of yeast paste. Lastly, load the adults into a population cage. On 24-hour intervals, replace the plate and check for the presence of eggs. Once sufficient eggs are laid, remove any dead flies from the plate and the remaining yeast paste from the collection.